So thank you for inviting me to this meeting. It's actually a real pleasure to be here. I'm not sure that Teresa knows this, but in 2006, 2007, whilst I was a postdoc with David Albertini at KUMC, Teresa actually came out and gave a talk at the Greenwood Symposium, and she spoke about oncofertility, and it was really the first time I'd heard of anything like this. And I remember sitting in the audience and thinking to myself that if I was ever lucky enough to have my own laboratory, then I'd really like to be able to contribute to this field. So here I am today, 12 years later, speaking to you, so it's a real privilege. So I'm going to talk about radiation and chemotherapy-induced cell death pathways in the oocyte. But I thought I might start with this slide, which really describes to you some of the work that my laboratory is doing in this area that I won't really have time to talk about all of this today, but if anyone wants to speak to me in the breaks, it'd be really great to talk to you. So first of all, we spend a lot of time in my laboratory characterising the impact of different types of cancer treatments on the follicle reserve. And we really focus on the genotoxic and DNA damaging agents, the common ones like radiation and cyclophosphamide and Cisplatin. But more recently, we're starting to look at some of the newer treatments like PARP inhibitors. There's very little known about the impact that PARP inhibitors like olaparib have on the ovary. So we're studying these in wild-type mouse, but also in BRCA1/2 mutant backgrounds. And we're also starting to take a look at some of the new immunotherapies that are really changing the face of cancer treatment. So we do most of these studies in mouse models, but we also have some access to cryopreserved ovarian tissue from Deb Gook at the Royal Women's in Melbourne. We spend a lot of time looking at mechanisms of primordial follicle depletion and in particular apoptotic regulators, and you'll hear about some of these today. Some of these studies have really led us to take a really closer look at DNA repair capacity in oocytes, genetic integrity in offspring, and also mitochondrial health. We've been investigating some new potential photoprotective agents such as NMN, which I won't be speaking about today, and also Puma inhibitors. And finally, we have quite a new research program in the lab that's starting to look at the impact of cancer treatments on the uterus. And there's actually not a lot of work out there on this area. And so I have a new postdoc in the lab who is really keen on investigating this. And she's been working with a PhD student to really try and nail down the effects of genotoxic and DNA damaging agents on the uterus that are distinct from those effects on the ovary. Okay, so we're all at this meeting because curative cancer treatments are a little bit like a double-edged sword. Early detection and improved treatments for cancer have resulted in dramatically improved survival rates. But at the same time, we know that these life-saving treatments can also have profoundly negative impacts on reproductive endocrine function and fertility. And that's because many of these treatments can cause collateral damage to the ovaries, and in particular, depletion of the follicular reserves. And if this depletion is severe enough, it can, of course, lead to infertility, but it can also lead to loss of ovarian endocrine function, and in particular, loss of estrogen production. And this can really have multi-systemic effects, including putting these patients at early increased risk of things like osteoporosis and heart disease. So the risk of ovarian damage is highly variable and it depends on the type of treatment, and we know that with the chemotherapy agents, we can divide these roughly into groups that pose a high risk, a moderate risk, or a low risk. And with the high risk categories, it's really the alkylators that we know are the most damaging. But we also know that there are a lot of new treatments that are now coming into use that we know very little about, that we really need to invest some time studying. For radiation therapy, it's all about the location, so radiation delivered into the vicinity of the ovary, such as to the pelvis, the abdomen, or spine, is going to be potentially damaging to the ovary, and of course, total body irradiation. But in addition to this, we know also that there are a number of contributing factors, including the age of the patient, the dose, and what combined treatments are used. So just to kind of summarise the scope of the problem that we have in Australia there are around 8,000 new cancer diagnoses each year in girls and premenopausal women, and this would obviously be proportionally larger in the US. And the risk of acute ovarian failure for all types of cancer and treatments is around 6%, and premature ovarian failure around 8%. But because of some of the variables that I've just talked about on the previous slide, we know that there are certain subsets of patients that are at much greater risk. For example, a young female receiving total body irradiation and an alkylator will almost certainly be infertile. Now, fortunately, as a result of much basic and clinical research over recent years, we now have some really good fertility preservation options available, and we'll hear about a lot of these over the next couple of days. The most well-established technique, of course, is the cryopreservation of oocytes, of embryos rather. So it involves harvesting mature oocytes and fertilising them, and then cryopreserving the embryos until the patient is ready to start a family. However, this technique does have some limitations that we need to think about. Firstly, it does require ovarian stimulation, which means it might not be suitable for some patients that need immediate treatment, or those with hormone-sensitive cancers, or for very young patients. It does require that the woman have a partner that she wants to have a child with, or be prepared to use donor sperm. Depending where you live, it can be very expensive. And whilst this is a very good option for preserving fertility, premature ovarian failure can still occur because the ovaries are being damaged. The second option is the cryopreservation of mature oocytes, and success rates have really improved dramatically in this area, particularly with the introduction of vitrification. One of the major benefits is that it does not require a partner or donor sperm. But it does come with some of the same limitations that I just described to you for the cryopreservation of embryos, and that it requires ovarian stimulation, it can be expensive, and the ovaries are still going to be damaged, so premature ovarian failure can still occur. So the third option is the cryopreservation of ovarian tissue, which is a surgical procedure where ovarian cortex is removed, it's frozen, and then later thawed and reimplanted at the original location or elsewhere in the body. The real benefit of this is that there is no delay. This can be performed immediately. And there theoretically is no lower age limit. So this is probably the only option that is available for very young girls. This has the potential to preserve both fertility and ovarian function, although there is some question over how long the lifespan of those individual grafts are. And a potential risk is that cancer cells may be reintroduced. So clearly we have some really good options available, but these treatments and options are not going to be suitable for all patients. And because of this, I think it's really important that we're investing in new areas of research to increase the fertility preservation options that are available. And active areas of research can really be broadly divided into three main groups. So the first one is the development of new methods for in vitro follicle growth and oocyte maturation. So here we have the potential to use that cryopreserved ovarian tissue, but avoid the risk of reintroducing cancer cells. And so this really requires the development of probably a multi-step, three-dimensional culture system that's able to support the complete in vitro growth of follicles and maturation of human oocytes. And we've actually had a lot of growth of this area in recent years. The second area of research is quite exciting, and that's this idea of being able to develop oocytes from stem cells. And here the idea is to be able to use embryonic stem cells or induced pluripotent stem cells, or in fact orgonial stem cells to make new oocytes. However, this strategy does actually require that we also are able to grow oocytes entirely in vitro. And the third strategy is the one that I'm going to talk about this morning, and that's the development of pharmacological ovo-protectants. So basically what I mean here is just the development of drugs that can actually protect the ovary from damage caused by chemotherapy and radiation. And so those of us working in the field spend quite a bit of time thinking about what the ideal pharmacological ovo-protectant might look like. And I've come up with 5 key properties. You may have some more to add to this list. But the first one is that it should be non-invasive. The second one is that it should be able to preserve the quantity and the quality of the ovarian reserve, protect fertility and ovarian endocrine function simultaneously. It should be able to confer protection against a range of chemotherapies, including those that we know are most damaging to the ovary, and that would be the alkylators and the platinum complexes. It should be suitable for females of all ages. And importantly, it should not compromise the effectiveness of the cancer treatment. But if we want to do this, then we really need a much better understanding of how different cancer treatments actually damage the ovary. So we know that DNA-damaging chemotherapy reduces the ovarian stockpile of these primordial follicles. You can see one here. It's got a large immature oocyte surrounded by a single layer of squamous granulosa cells. Primordial follicles are the most abundant form of oocyte found in the ovary. You can think of them as a storage unit for the female germline. And importantly, they're the precursors of all these mature hormone-producing follicles and the source of all oocytes for ovulation. But they're a bit unusual. They exist in this unique stasis where they've initiated meiosis and then arrested partway through the first meiotic prophase at depleting. And they can remain arrested for many decades. Also, they're non-dividing. And despite some controversy in this area, it's still widely accepted that a baby girl is born with all of the primordial follicles that she will ever have, and that this supply can't be replaced even if it's prematurely depleted. And finally, primordial follicles are extremely long-lived. So of course, an oocyte ovulated by a woman in her 40s is derived from a primordial follicle laid down in her ovaries before she was born. And because of this, we think there must be very good quality control mechanisms in the ovary to protect those oocytes. Because of all these things, the number and quality of primordial follicles influence the length of the fertile lifespan, the age at which menopause occurs, and can also influence offspring health. So then how are primordial follicles actually depleted following chemotherapy? And so one of the more recent schools of thought is that follicle depletion occurs through a process called burnout. And I'm just going to briefly describe this process to you here. And this concept was really arising from the fact that primordial follicles are non-proliferative. They don't divide. And so it was proposed that therefore they can't be damaged by chemotherapy agents, which typically more specifically target proliferating cells. And secondly, it's been very hard to find dying primordial follicles in the ovary after chemotherapy, and lots of labs have experienced this. And so it was proposed that what's really happening is that chemotherapy causes the death of secondary follicles, which of course have a lot of proliferating granulosa cells. Now these secondary follicles have a role in the ovary in secreting factors that control the rate at which primordial follicles activate. And so in the context of chemotherapy, the depletion of the secondary follicles would subsequently lead to an accelerated activation of the primordial follicle pool, which would ultimately lead to depletion of the primordial follicle reserve. However, my lab has actually been investigating the alternative hypothesis, and that's that chemotherapy can in fact damage primordial follicle oocytes directly, leading to their apoptosis. And there are a couple of reasons why we think this. Firstly, the drugs that are most likely to impact on fertility are those that are non-cell cycle specific. So they can potentially damage oocytes which are non-proliferative. And secondly, if you look through the literature, there's actually some very nice and elegant older studies which do suggest that drugs like cyclophosphamide can directly kill primordial follicle oocytes. And so trying to understand the mechanisms that underlie the depletion of primordial follicles really is not just an academic endeavour. It's really important because by understanding the mechanism, that's going to really inform how we go about designing fertility preservation agents. So if burnout depletes primordial follicles, then effective fertility preservation agents will really need to prevent primordial follicle activation. Alternatively, if chemotherapy directly damages primordial follicle oocytes, then effective fertility preservation agents will need to prevent primordial follicle death and support oocytes to repair their DNA damage. Or it could be that both options are required. So my lab spends a lot of time thinking about apoptosis and investigating apoptosis, which we all know is a form of programmed cell death that's really essential for development and homeostasis. But under certain circumstances, it can also be highly detrimental. It does have this really distinct morphology where both the cell and the nucleus fragments and then forms into these apoptotic bodies. And this is important because this is what avoids the inflammatory response. So there are two main pathways for apoptosis that cells can use to die, and the one that's relevant for this talk today is the intrinsic apoptosis pathway, which I'm showing to you here, and the details are not super important. But what you do need to know is that this is the apoptotic pathway that is triggered by DNA damage. It's then controlled by a balance between the survival proteins, which are shown in this figure in blue, and the death-promoting proteins, which are shown in this figure in pink. And so what happens is when the pathway is tipped in the favour of the death-promoting proteins, you get activation of BAX and BAC, you get mitochondrial damage, and ultimately activation of caspases and cell death. Now what's really interesting is that a number of elegant studies from a few years ago now have shown that the inhibition of caspases is actually insufficient to cause cell death, to prevent cell death. If you want to prevent cell death, then you really have to prevent the activation of BAX and BAC. And one of the ways that you can do this is by altering the balance between the cell death and the cell survival proteins at the top of this cascade. And so we wondered if we could actually prevent primordial follicle depletion caused by chemotherapy, if we could block this process, if we can block activation of BAX and BAC. And we focused in on a pro-death or pro-apoptotic protein called Puma. And we did this because Puma is a BH3-only pro-apoptotic protein at the top of this cascade. It's known to be triggered in response to genotoxic stress. In somatic cells, it's transcriptionally induced by P53, and we showed a number of years ago now that in oocytes, it's actually transcriptionally induced by P63. And you'll hear a little bit more about P63 later in the day with Soo-Yoon Kim speaking. And so a PhD student in the lab 3 years ago now started investigating these three questions. Does eliminating Puma protect the ovarian reserve against DNA-damaging chemotherapy? And would this, in fact, preserve fertility? And finally, what are the mechanisms of chemotherapy-induced follicle loss? And so she designed a very simple series of experiments to investigate this. She used adult female mice that were either wild type, deficient in Puma, or deficient in P63, which is that upstream transcriptional activator of Puma. These mice were treated with saline, cyclophosphamide, or cisplatin. She harvested ovaries 8 or 24 hours later to look at mechanisms of follicle loss, and to do this she counted follicles, she looked at primordial follicle morphology, she looked at markers for DNA damage and apoptosis. She also harvested ovaries 5 days later and looked for follicle survival by doing follicle quantification, and again looking at primordial follicle morphology. And finally she did this long-term fertility study where she looked at a number of parameters, including looking at the duration of fertility, which is probably the most important marker of fertility in mice in this type of study. She looked at total number of pups, litter number and size, and also offspring health. And I'm just going to show you a couple of the highlights from this study. So this graph shows follicle survival at 5 days following cisplatin or cyclophosphamide. And if you just look at the white bars first, this is for wild-type females, you can see that both cisplatin and cyclophosphamide dramatically reduce the size of the primordial follicle reserve. But what's really interesting from these studies is that we found that loss of Puma, shown in the blue bars, loss of Puma alone is sufficient to rescue all of the primordial follicles from apoptotic depletion. Interestingly, we found that loss of P63 is sufficient to rescue all of the primordial follicles from cisplatin-induced loss, but it's actually only partially protective against cyclophosphamide. And so this very simple study told us that Puma is in fact the key mediator of primordial follicle loss after cisplatin and cyclophosphamide. And that cyclophosphamide-induced primordial follicle loss occurs independently or largely independently of P63. So actually cisplatin and cyclophosphamide induce Puma probably through different pathways. So we were able to rescue all of the primordial follicles from loss caused by cisplatin and cyclophosphamide in the Puma knockout females, but what actually did these primordial follicles that survived look like? Well, if you look at the middle panel on this particular figure for the Puma knockout females and saline, cyclophosphamide and cisplatin treated animals, you can see that they actually look morphologically normal. They don't look any different after cisplatin or cyclophosphamide treatment. But morphology is really not good enough indicator of oocyte quality. And that's because any unrepaired DNA damage that has occurred really has this potential to induce infertility or lead to genetic abnormalities which could in fact be propagated into the offspring. So what's the quality of these surviving oocytes? Can these rescued oocytes actually give rise to sustained fertility in normal offspring? And so that's where our fertility studies came in. And this slide here shows you the data for the wild-type females and just a couple of the parameters. And what you can see is that the fertile lifespan is shortened in the cyclophosphamide and cisplatin treated female mice. So it's around 175 days shorter after cyclophosphamide, which is about half the normal expected lifespan, and about 95 days shorter after cisplatin. There's also a reduction in the number of litters and pups after cyclophosphamide, but this is not so obvious with the cisplatin treatment. But importantly, all of these fertility parameters were completely preserved in the Puma knockout females. So these rescued oocytes are in fact of sufficient quality to support fertility and produce healthy offspring. So we know that loss of Puma can protect oocytes or prevent the oocytes and primordial follicles from undergoing apoptosis and dying. And we know that these oocytes can then go on to produce healthy offspring. But we really wanted to investigate further the mechanisms of chemotherapy-induced follicle loss. And so the data that I'm showing you here is follicle counts at 24 hours post-chemotherapy. The previous data was for 5 days, and this is for 24 hours after. And what we found was that these primordial follicles are actually depleted really quickly. They're depleted within 24 hours. And not only could we see a depletion of primordial follicles, we can also at this time point see a depletion in the transitional and the primary follicles. And when we look at the secondary and antral follicles, we also see no evidence for any increase in any other follicle class. So if burnout were occurring at this early time point, you would expect the depletion in primordial follicles to be happening at the same time as there is an increase in transitional primary or secondary follicles. And we simply don't see it in this model. So in this particular model that we're using here, primordial follicle depletion cannot actually be attributed to burnout. And so we wanted to investigate the burnout theory a little more closely. So we asked a slightly different question. We asked what would happen if we specifically deplete the growing follicle population. And so we did another experiment using adult wild-type female mice, and these mice were treated with saline, or a chemotherapy agent known as 5-FU. So this is an anti-metabolite chemotherapy that will specifically induce apoptosis in proliferating cells, not in non-proliferative cells. So these ovaries were harvested 12 hours and 7 days after this treatment, and we found that, as we expected, there was a significant amount of apoptosis in the granulosa cells of secondary follicles, which we could measure very clearly by caspase staining or tunnel staining. And there was an increase in the percentage of secondary follicles that were undergoing apoptosis, and an increase in the number of secondary follicles and antral follicles that were atretic. But despite the depletion in the number of growing follicles, we were unable to detect a depletion in the number of primordial or primary follicles. So atresia and loss of secondary follicles and antral follicles in this model was not sufficient to actually induce a depletion of the primordial follicle reserve. So we thought, well, perhaps this was because we gave a single dose. What happens if we repeatedly deplete secondary follicle populations? So we did a similar experiment, but this time we treated the mice with saline or 3 weekly doses of 5-FU. Ovaries were harvested 6, 12 and 36 hours after this time point. We couldn't leave these mice any longer because they're actually quite sick after this treatment. We were able to detect a depletion in the secondary follicles, a depletion in the antral follicles, and an increase in atretic follicles. In this particular study, we also measured AMH levels, and across the treatment period, we could very clearly detect a depletion in AMH. But despite this, we found that repeated depletion of growing follicles on its own was not sufficient to deplete primordial follicles. So whilst we do see evidence of secondary follicle atresia after cyclophosphamide and cisplatin treatment, we don't think this is sufficient to cause the rapid depletion of primordial follicles that we see after these treatments. So we then decided to ask this question: do cisplatin and cyclophosphamide actually directly damage and kill oocytes in primordial follicles? And so we investigated the cellular targets of these drugs in wild-type ovaries at an early time point, so 8 and 24 hours after treatment. And so in these images, the blue is DNA and the red is gamma-H2AX. Gamma-H2AX, for this particular study, is a marker of double-strand break, so a marker of DNA damage. And I appreciate it might be quite difficult to see in this room, but what I'm showing here is that in the saline-treated ovaries, there is no evidence of gamma H2AX in primordial, transitional, or primary follicles, or in fact secondary or antral follicles. But after cyclophosphamide treatment and after cisplatin treatment, there is very clear evidence of DNA damage in primordial follicle oocytes and in transitional follicles and primary follicles. Interestingly, we found that there was a significant amount of DNA damage in the proliferating granulosa cells of secondary and antral follicles, but we didn't detect gamma H2AX in the oocytes at those later stages. So this panel really shows us that these agents are in fact able to directly damage primordial follicle oocytes, but do these oocytes go on and die? So we spent a lot of time looking histologically at ovaries and staining for markers of apoptosis, and we could in fact find quite clear evidence, morphological evidence of oocytes undergoing apoptosis 8 and 24 hours after these treatments. And this is just an example of the types of things that we would see. So the first panel after saline is what a normal primordial follicle oocyte looks like. But after cyclophosphamide treatment, you see a lot of these oocytes with a jelly bean-shaped nucleus, which is a nucleus on the way to condensing and becoming apoptotic. We would see fully condensed and fragmented nuclei, and these oocytes would in fact stain positive for tunnel, meaning that they were undergoing apoptosis. And then if you look 5 days later, we would see a lot of these empty follicles in which the oocyte has been destroyed, but the granulosa cells are remaining, and these remaining granulosa cells still stain positive for a granulosa cell-specific transcription factor, FOXL2. So they do in fact die. So just to summarise this part of the talk, I've shown you that cisplatin and cyclophosphamide do induce DNA damage and oocyte death in primordial follicles. And under the conditions that we've used in this particular study, burnout does not appear to be a primary mechanism of follicle depletion. And so we think that effective pharmacological fertility preservation strategies should really include inhibition of apoptosis, and perhaps this should be done in combination with inhibitors of activation. We've shown that PUMA is a critical apoptotic trigger in ovarian reserve depletion following cyclophosphamide and cisplatin. In a previous study, which I haven't talked about today, we've also shown that it's the key apoptotic regulator after irradiation also. So this means that the elimination of a single protein is really sufficient to protect oocytes from what are highly gonadotoxic treatments. In this instance, fertility is completely preserved, and the offspring do appear quite healthy. And what we found quite interesting in the irradiation study, which I haven't talked about, is that you only really need to rescue quite a low number of primordial follicles. We found that we could rescue only around 12% of primordial follicles, and that was sufficient to have really good fertility protection. So our data so far suggests that these oocytes are capable of repairing their DNA, and I'm going to come to this again in a moment. We've also done some studies to look at the surviving mitochondria in these oocytes and found them to be functionally normal. And what's really exciting for us is that we've done all of these studies so far in a genetic model, so Puma knockout females. But earlier this year, there were 2 Puma inhibitors have now been characterised in the literature. And these small molecule inhibitors of Puma are in fact available, and they do not compromise chemotherapy efficacy in animal models. So because of this, we think that Puma inhibitors may in fact represent an exciting new fertility preservation option. And certainly our results in our Puma knockout females suggest that Puma inhibitors may meet many of these ideal ovo-protectant criteria. And so we're now just initiating our studies to test these inhibitors in our mouse model. And so just to finish this part of the talk, I wanted to leave you with this thought. So the more we learn about the mechanisms underlying cancer treatment-induced follicle depletion, the more likely we are to be able to develop effective strategies and identify novel targets for preservation of the ovarian reserve. But understanding their biology is really essential if we're going to do a good job of this. All right, so I just wanted to switch gears a bit now and talk to you a little bit more about DNA repair within oocytes. So we know that oocytes and primordial follicles are really sensitive to DNA damage. They're more sensitive than embryonic oocytes, they're more sensitive than somatic cells, and they're in fact more sensitive than the oocytes in growing follicles. So embryonic oocytes will actually withstand hundreds of double-strand breaks that are induced during meiosis. but in fact just a few double-strand breaks in the oocytes of primordial follicles will trigger apoptosis. And it's actually been proposed that oocytes have very sensitive mechanisms for the detection and elimination of cells with DNA damage, and that this might be necessary to ensure that oocyte quality is maintained and protect against the introduction of germline mutations. But all of the data that I've just shown you suggests that oocytes in primordial follicles are in fact capable of repairing their DNA. And when we were thinking about the results of our data, we went into the literature, but we really couldn't find very much information about DNA repair in primordial follicle oocytes. And so Jessica Stringer, a postdoc in my laboratory, has spent the last nearly 3 years now investigating DNA repair in primordial follicle oocytes. And she's really been focused on these 3 questions. Do oocytes activate a DNA repair response? And if so, which pathway do they use to repair their double-strand breaks? And what's the capacity for DNA repair in these oocytes? And by that I mean how much DNA damage can they sustain and repair in order to protect offspring health? And finally, is apoptosis actually essential for quality control in the female germline? So in other words, how efficient is that DNA repair at restoring oocyte quality? And to do these studies, she's been using similar mouse models to what I've just been talking about. So wild-type animals, Puma knockouts, and P63 knockouts. And really the Puma and the P63 knockouts were found to be essential for this study, and that's because these oocytes can't undergo apoptosis. So it gives us time to be able to determine whether they can actually initiate repair processes. So she's been using a range of mice. She's been using neonatal mice, young adult mice, and reproductively aged mice. And that's because we think there might be a change in the ability to repair DNA across the lifespan, with potentially reproductively aged females being less able to repair their DNA damage. And she's used an irradiation model for the study, not a chemotherapy model, starting with a very low dose of a 0.1 gray through to a 7 gray dose. And in these studies, she's been characterising the temporal DNA repair response in primordial follicle oocytes, focusing on the 2 different repair pathways that are involved in repairing double-strand breaks. So that's homologous recombination and non-homologous end joining. She's been looking at the ability of these surviving oocytes to mature and to produce pre-implantation embryos and offspring. She's done a lot of studies evaluating offspring health and also doing genome sequencing of the offspring to look for genetic mutations. And so from her studies and from some of our earlier work, we now know that the DNA damage response is in fact activated in primordial follicles. So this panel shows you DNA in red and phosphorylated ATM in green. So phosphorylated ATM is a global regulator of the DNA damage response, and normal healthy oocytes have very little phosphorylated ATM. But ATM becomes rapidly phosphorylated after irradiation, and you can see it clearly in this panel. Now, one of the jobs of phosphorylated ATM is then to go on and coordinate the DNA repair response. And one of the ways that it does this is by phosphorylating histone H2AX, which becomes known as gamma H2AX. Gamma H2AX then forms foci at the sites of DNA damage, and these foci act like platforms for the recruitment of DNA repair proteins. And so you can really effectively use gamma H2AX to monitor both the induction of repair and then the subsequent resolution of all of the DNA damage. And so that's what we've done here, where the DNA is in blue and the gamma H2AX is in red. We find that normal healthy oocytes have no gamma H2AX foci, but these foci can be readily observed 3 hours after radiation at 0.45 gray, and this is the dose response here at 10-fold higher gray dose. You can see that there's much more gamma H2AX present. Now, in our Puma knockout females, if we stop these oocytes from dying and we look 5 days later, we can very, very clearly see that all of these gamma H2AX foci are completely resolved, indicating that DNA repair is occurring. And so Jess has really taken these studies a lot further and done many, many more over the past 2.5 years. And she's been able to show that primordial follicle oocytes very rapidly detect and respond to DNA damage. And this data that I'm showing you here is from the P63 knockout females. And this is the percentage of positive oocytes for 3 markers of the DNA damage response, 30 minutes, 3 hours, and 6 hours after radiation. And these proteins can all be detected in nearly all of the oocytes, just 30 minutes after gamma radiation. The timing here is dose dependent, I will add. And if we just look at the gamma H2AX, we can see that 3 hours after irradiation at a 0.45 gray dose, around 90 to almost 100% of the oocytes will actually express gamma H2AX foci, and these foci are resolved in approximately 50% of the oocytes at 24 hours, meaning that repair has been completed. And in thinking about which particular pathways an oocyte might use to repair itself, we've looked at a lot of the different pathway components, and I'm just showing you 2 of the key ones here. So RAD51 is associated with homologous recombination. DNA protein kinase CS is involved with non-homologous end joining, which is actually the DNA repair response that is typically used by somatic cells, but it's error-prone, so it can create mutations. And what we found was that around 90% of the oocytes have RAD51 foci after DNA damage. And this indicates to us that homologous recombination is probably the pathway of choice. And this is really expected because the oocytes, the primordial follicle oocytes, have already replicated their DNA, which means they have a template available to be able to undertake homologous recombination repair. What was interesting to us is we did find around 10% of the oocytes do express non-homologous end-joining proteins, suggesting that under certain circumstances, oocytes may be undertaking this error-prone form of DNA repair. So we know that oocytes initiate a DNA damage response and they localise DNA repair proteins to the sites of DNA damage. But how good is that repair really? To answer that question, we've done a lot of studies looking at the ability of oocytes to complete meiosis and complete subsequent embryonic development. And interestingly, what we found was that after the 0.45 gray dose of oocytes, we got the same number of primordial follicles. We were actually able to obtain fewer oocytes after superovulation. And we're not entirely sure why that is. But importantly, all of the oocytes that we were able to harvest were morphologically normal and had completed meiosis normally. And then when we did natural matings with these mice, we found that these mature oocytes were fertilised at a normal rate and that these oocytes were also capable of supporting normal rates of pre-implantation embryonic development. But what about offspring? We get normal pre-implantation embryonic development, but do we get normal offspring? And so these graphs just show you the number of litters and the number of pups in our wild type and our P63 animals, and just focusing in on the 0.45 gray dose. In the wild types, you'll see that there are no litters and no pups. These animals are infertile because all of the primordial follicles have been destroyed. In the P63 knockout females, we were able to rescue the primordial follicles from a radiation-induced death, where we've been able to show that there is an induction of a DNA damage response and induction of a DNA repair response. These females do in fact have normal numbers of litters and normal numbers of pups. We've gone on to look at all of the histopathology from all of the offspring from these animals, and that includes all of their organs, and we found no abnormal histopathology. And we're currently in the process of doing whole genome sequencing on these offspring, and we've got about 8 animals done, and we're in the process of doing the remaining now, so hopefully I'll have that data for you soon. So just to summarise this part of the talk, we've been able to show that oocytes from Puma and P63 mice withstand a higher level of DNA damage than wild-type mice, and we can really exploit this to begin to examine DNA repair capacity. We've been able to show that the DNA damage response is in fact activated within 30 minutes of DNA damage, and the DNA repair response is activated within 3 hours. And that oocytes can in fact repair their DNA damage within 24 hours, which is pretty quick. It appears that homologous recombination is the pathway of choice, and this is important because this is a high fidelity repair pathway, and that this repair is sufficient for normal embryonic development and can support offspring health. So our offspring from the irradiated Puma and P63 mice are healthy, despite the fact that their oocytes have sustained a really large amount of DNA damage, certainly enough DNA damage to induce apoptosis. And so for my final slide today, I just wanted to conclude with this thought. So apoptosis may not be essential for quality control in the female germline as we had previously thought. Despite their propensity for apoptosis, primordial follicle oocytes do in fact have the capacity for highly efficient DNA repair. And this DNA repair is in fact sufficient to restore oocyte quality and ensure offspring health. And I think this is really good news for us, for those of us that are really interested in developing these new ovo-protectant agents that work by preventing the death of these oocytes. So I just need to finish with a couple of acknowledgements here. So all of the work that I spoke to you about today was done by members of my laboratory, in particular Jessica Stringer, who did all of the DNA repair work, and Quinn Nguyen, who is an MD PhD student who is just finishing up her PhD now, who did all of the work with the chemotherapy and the P63 and Puma knockout females. So thank you.